Good morning, students. In this class, we will study about cell cycle and cell division. So, cell cycle. Cell cycle means a series of events or changes take place between two cell divisions. Here it is cell division, cell cycle. Cell cycle can be divided into two major events, and these are interphase, which is also known as non-dividing phase, because here division does not take place. And second is M phase, which is also known as mitosis. Here we can see interphase. It can be divided into two phases, here G1 phase, S phase, G2 phase. So again, interphase can be divided into G1 phase, S phase and G2 phase. Here G1 phase means first growth phase. Second S phase means synthesis phase. And number 3, G2 phase means second growth phase. So here interphase, G1 phase, S phase and G2 phase, these are the three subphases of interphase. The longest subphase of interphase is the G1 phase. It takes more time than S phase and the G2 phase. And again, S phase, which is known as synthesis phase, it is the smallest subphase of interphase. Now, what are the changes taking place during G1 phase? During G1 phase, a number of changes taking place, and these are: number 1, synthesis of RNA. As you know, RNA means ribonucleic acid, which is a type of nucleic acid, RNA and proteins. So more proteins and RNA are needed for the proper growth of the daughter cells, because when a cell divides into two parts, the volume of the cytoplasm as well as different types of cell organelles become reduced. So it is necessary to synthesize more RNA as well as proteins in the during the first growth phase, and here synthesis of RNA and proteins takes place. Number 2, chloroplast in plant cell. Chloroplasts in plant cell and mitochondria in both the type of cells. Mitochondria in both the type of cells means in both plant and animal cells. Chloroplast in plant cell and mitochondria in both plant cell and animal cells duplicate. Here duplication means formation of new chromosome, chloroplasts as well as mitochondria. So synthesis of RNA and proteins, it is the first event. Second, chloroplasts and mitochondria duplicate. Now G2 phase, S phase S phase, which is the synthesis phase. Synthesis phase. During the synthesis phase, synthesis of DNA. Synthesis of DNA means duplication or replication. Duplication or replication of DNA takes place. It is the first event. Second event, chromosomes become duplicated. So these are the two major events take place during S phase. Now the third, which is known as second growth phase. Second growth phase or G2 phase. Second growth phase. During second growth phase, again more RNA and proteins form or synthesize. And number two, duplication of chloroplasts and mitochondria take place. So these are the two events take place in G2 phase, which were started in G1 phase. So after G1 phase, S phase means duplication of chromosomes as well as DNA take place, and in again G2 phase, more RNA and proteins form or synthesize, and duplication of chloroplast in plant cells and mitochondria in plant as well as animal cells take place. So these are the major changes or events take place during G2 phase. Now sometimes G0 phase means no growth phase. No growth phase. Here G0, when a cell becomes damaged severely and it cannot repair itself for S phase, then it withdraws and comes in G0 phase, means no growth phase. Actually it is resting phase. Resting phase, and in this phase the cell which is damaged severely try to repair itself, try to make itself for the next subphase, which is S phase. And in this phase, here there may be two conditions. First, after G1 phase, the cell undergoes in S phase. Otherwise, it can withdraws and comes in G0 phase for rest and for preparation for the second subphase, which is known as S phase. So here resting phase, here it is known as resting phase because there is no changes taking place, resting phase. But here no changes taking place, but it prepares itself for S phase or undergoing in the next subphases. Here we can see interphase and mitosis or M phase. These are the two steps involved in cell cycle. This one is the cell cycle. Cell cycle, interphase, G1 phase, S phase, G2 phase, and sometimes G0 phase. These are the divisions of the interphase. Here G1 phase is the longest and S phase is the smallest subphase. Now mitosis or M phase. Mitosis can be divided into 4 subphases: Prophase, Metaphase, Anaphase, Telophase. These are the 4 phases which are collectively known as karyokinesis. So karyokinesis is the division of nucleus, or you can say it is the division of karyokinesis, division of chromosome or nucleus. So it is the division of nucleus. As you know, nucleus contains chromosomes, and actually it is division of chromosomes. Chromosomes become duplicated, and now it divides, splits into 2 chromatids, 2 new chromosomes, and this is known as karyokinesis or division of nucleus. Here we can see prophase is the first phase, metaphase, then anaphase, and finally telophase. Now what are the changes taking place in these phases? Now first is prophase. Prophase means, pro means early. So it is the first phase of karyokinesis, and therefore it is known as prophase. A number of changes taking place here, and these changes are very important. So here we can compare the changes taking place during prophase. Here it is interphase, now it is cell membrane, nuclear membrane, centrosome, nucleolus, and these are chromatin fibers. So during interphase, cell membrane, centrosome, nuclear membrane, nucleolus, and chromatin fibers, these are the major parts of the cell. Here centrosome is present, and also cell wall is not present. Therefore, we can say it is an animal cell. Now, what are the changes taking place in prophase? So, now prophase, during prophase, what are the changes taking place? We are discussing. At first, diagram we have to draw. Prophase is the longest phase of mitosis, and therefore conventionally we can divide it into 2 parts, early prophase and late prophase, for easy study. Now here it is centrosome. Centrosome splits into 2 centrioles, and they move apart from each other. They move towards the opposite poles. So here it is centrosome. This one is the nuclear membrane, here it is shell membrane. Now inside the nuclear membrane, nucleolus is present, and chromosomes are now start to visible, because here chromatin threads are found which are thread-like structures, and these thread-like structures cannot be seen properly. Means these are invisible. Now during early prophase, these chromatin fibers become distinct, and when we can see these chromosomes properly. Now during late prophase, nuclear membrane and nucleolus disappear. So it is nuclear membrane which is disappearing, and also the nucleolus, and chromosomes become visible or distinct. So here these are disappearing nuclear membrane. Now centrosome, which are located at the opposite poles, and these are radiating rays. Radiating rays means the rays which surrounds centriole are known as radiating rays. These are visible chromosomes. So chromosomes are visible here. Centrioles are located at the opposite poles. Chromatids as well as chromosomes become visible, and from the centrioles, opposite centrioles, a number of spindle fibers form, and these are attached with centromere of each chromosome. Spindle fibers are made up of protein, and they have elasticity. They can contract and relax. As a result, distribution of chromosomes inside the nucleus take place due to contraction and relaxation of spindle fibers. In this way, here distribution of chromosomes take place. Chromosomes are scattered inside the nucleus. Now nuclear membrane and nucleolus disappear. So chromosomes are scattered in the middle part of the cell, and in the peripheral part all the cell organelles are located as well as the cytoplasm. Here it needs more space inside the cell. That's why in the center, in the middle part of the cell, chromosomes is scattered, and in the peripheral or marginal part the cell organelles are located. So here between prophase and between early prophase and late prophase, a number of differences we can see. Number one, in the early prophase, centrosome splits into two parts and move towards the opposite pole. But in the late prophase, we can see centrioles are located at the opposite poles. Now number two, nuclear membrane and nucleolus start disappearing. But in case of late prophase, these are disappeared. So it is the second major difference. Now what are the changes taking place during prophase? Number one, nuclear membrane and nucleolus disappear. Number two, second point here, chromosomes become distinct and visible. Number 3, centrosome splits into centrioles and move towards opposite poles. And spindle fibers are attached with centromere. Here this structure is known as centromere. These are the spindle fibers here. These are spindle fibers. And aster rays, which are radiating rays which surrounds the centrioles here, and these are the chromosomes. So chromosomes become distinct or visible. Number 2, centrioles are located at opposite poles. Nuclear membrane and nucleolus disappear. Chromosomes become visible. Spindle fibers are attached with centromere of each chromosome, and centrioles form aster rays. So these are the events taking place during prophase. Now the second phase, which is known as metaphase. Here it is metaphase. During metaphase, centrosome, now centrioles, are located at opposite poles, and all the chromosomes are located at middle of the cell or nucleus, which is known as equatorial plane. It is known as equatorial plane or plate, and here chromosomes are visible or become distinct, and it is the phase where chromosomes can be studied properly. Now these are the spindle fiber. Here it is centrosome or centriole. These are radiating rays which are known as aster rays. Here chromosomes, and these are the 4 chromosomes located at the plane, at the same plane, which is known as equatorial plane or equator. What are the changes taking place during metaphase? Number 1 is centromere are attached with spindle fiber. Number 2, which is the most important event of metaphase, is here chromosomes lie at equatorial plane. Chromosomes lie here at equatorial plane, and centromere of each of the chromosome are attached with spindle fiber. So here these are the changes taking place. In the next phase, which is known as Anaphase, during Anaphase Again a number of changes taking place. It is the smallest phase of mitosis cell division or mitosis, whereas prophase is the longest phase. So during anaphase, the significant feature of anaphase is the chromosomes split from centromere. As you know, centromere is the central point or point of attachment of two sister chromatids. So when centromere splits, then it forms V-shaped structure because new chromosomes are drawing towards the opposite poles. So here it is centromere and these are the two sister chromatids. Suppose here these are attached with spindle fiber, and when in the spindle fiber contraction takes place, then centromere splits into two parts and these are moving towards the opposite poles. As a result, they form V-shaped structure. So after splitting of the centromere, the new parts, means the new chromatids, are moving towards the opposite pole. That's why they look like V-shaped structure. And here you can see there where 4 chromosomes are located in metaphase, and now 4 sets of new chromatids or chromosomes are seen here. So duplication takes place in S phase, duplication of chromosomes takes place in S phase of interphase, but now here it splits into two parts, and each here it is single chromosome and these are the duplicated chromosome. It is non-duplicated chromosome. So when these duplicated chromosomes split into two parts, then it forms two non-duplicated chromosomes or new chromatids. So these are moving towards the opposite pole, and here they move towards the opposite pole due to contraction in the spindle fiber. And finally, at the end of anaphase, a cleavage furrow is formed which move towards the opposite poles. And here chromosomes now lie near the centriole or opposite pole, so here we can say it is early anaphase, and here it is late anaphase. In the early anaphase, chromosomes after splitting are moving towards the opposite poles. In the late anaphase, you can see chromosomes after splitting move towards the opposite poles and now lie near the opposite poles, and a cleavage furrow formation starts during late anaphase. So here a number of changes taking place again. Number one is, which is the most significant, each chromosomes split and move towards the opposite poles. Number 1 and number 2, during late Anaphase, cleavage furrow formation starts. So here these are the two events or changes taking place. Now the last phase is known as Telophase. During Telophase, furrow formation depends towards the center. Nuclear membrane and nucleolus start reappearing. Chromosomes now become invisible, and the cleavage formation, which starts at Anaphase, now deepens towards the center. And now finally it splits into two parts. Now these are the two daughter cells. So from one mother cell or parent cell, two daughter cells form after telophase, and this division of the cytoplasm is known as cytokinesis. Cytokinesis. It is the telophase, and now this one is the cytokinesis. So cytokinesis is the division of shell, or you can say division of cytoplasm. Division of cytoplasm. Cytoplasm divides, and finally it forms two daughter cells. So in this way, during the cell cycle, there are two steps, two major steps. First is interphase and second is M-phase or mitosis. During interphase, G1 phase, G2 S phase and G2 phase take place, and sometimes G0 phase. Now in the M phase or mitosis phase, there are four subphases. These are Prophase, Metaphase, Anaphase, Telophase, and these are collectively known as karyokinesis, means division of chromosomes or nucleus, and after it cytokinesis takes place. Cytokinesis is the division of cytoplasm, and after cytokinesis, here you can say it is cytokinesis. Here cytokinesis takes place, and after cytokinesis, one cell divides to form two daughter cells, in which number of chromosomes are similar. In the next class, we will discuss about importance or significance of mitosis cell division, differences between mitosis and meiosis, and also meiosis cell division. Thank you, students.